Hello, we are Group Q. Welcome to Can We Restart the Heart?
The research presented in this video focuses on human progenitor cells. Progenitor cells are similar to stem cells, but more specific because they are pushed towards differentiating into a certain type of cell. They can only replicate a finite number of times, whereas stem cells can keep replicating forever. There are many examples of progenitor cells in the body, including myosatellite cells, which are found in mature muscle and give rise to either more satellite cells or differentiated skeletal muscle, endothelial progenitor cells, which are involved in the regeneration of the endothelial lining of blood vessels and are important in wound healing, and cardiac progenitor cells, which will be the focus of this video. In previous times, the existence of cardiac progenitor cells was not known because it was believed that the heart was postmitotic, meaning that the heart did not continue mitosis or cell division after fetal development was complete. Now it is known that human cardiac progenitor cells, or HCPCs, have the ability to form the three types of cells in the myocardium: myocytes, smooth muscle, and endothelial vascular cells. The ability of HCPCs to maintain cardiac repair is influenced by many factors, including genetics, heart load, diseases, medication, and aging. This cartoon illustrates the process of myocardial renewal and how the heart responds to pathologic overload. Certain niches in the heart contain stem cells and cardiac progenitor cells. The progenitor cells are actively proliferating, and after they become activated, they differentiate into the three myocardial cell types previously mentioned. The relationship between cardiac regeneration and the enzyme PIM1 will be the main focus of this research and thus this video. The gene PIM1 is a proto-oncogene, meaning that the gene has the potential to cause cancer when mutated or expressed at high levels. The enzyme PIM1 is a threonine-serine kinase, which means that it phosphorylates the hydroxyl group of specific threonine or serine residues. It is involved in cell cycle progression, apoptosis, cytokine signaling, and transcriptional activation. It is implicated in many human cancers such as prostate cancer, certain types of leukemia, Burkitt's lymphoma, and oral cancers.
Figure 1 shows the different human cardiac progenitor cells normally. The bars in black are the fetal human cardiac progenitor cells. The striped bars represent the medium growth rate adult human cardiac progenitor cells, and the gray bars are the slow-growing adult human cardiac progenitor cells. Fetal human cardiac progenitor cells showed the highest levels of proliferation, as shown in graph A, survival, as shown in graph C, and telomere preservation, as shown in graph E. They also show the highest levels of PIM1, as shown in graphs D and G. Graph B shows that the adult human cardiac progenitor cells had a higher doubling time than the fetal human cardiac progenitor cells. Figure 4 shows the adult human cardiac progenitor cells that have been altered with nuclear-targeted PIM1 compared to the fetal cardiac progenitor cells. The PIM1 modification showed significantly higher telomere lengths than the adult human cardiac progenitor cells without the modification, as shown in graph A. P53 and P16 mRNAs had lower levels with the PIM1 modification. These mRNAs are associated with senescence. The NS mRNA had lower levels with the PIM1 modification. NS antagonizes senescence. Graph A in Figure 6 shows the increased proliferation rate with the PIM1 modification. Graph B further explains this by showing the shortened doubling times.
The modifications due to PIM1 turn back time for the human cardiac progenitor cells. By elongating the telomeres, increasing proliferative potential, and decreasing susceptibility to apoptosis, this gene/enzyme extends myocardial use. This study demonstrates for the first time that the intracellular targeting of PIM1 regulates the cardioprotective properties of the heart by enhancing proliferation and survival of HCPCs. The overexpression of PIM1 in the nucleus of the HCPC causes an increased proliferation and supports antagonism of HCPC senescence. PIM1 expression in the nucleus would be more impactful for a person whose HCPCs display more pronounced senescent phenotype, but are also resistant to cell death. This study demonstrates that PIM1 localization preferentially enhances proliferation, survival, and useful properties of aged HCPCs dependent upon intracellular localization. Targeting PIM1 to the mitochondria has proven to be very effective because it supports maintenance of energy metabolism shown by the increase in ATP production. By locating the PIM1 expression in the mitochondria, it increases protein expression of cyclins and CDKs more effectively than the previously tested PIM wild type, which is essential because cyclins and CDKs regulate the cell cycle progression. Due to the high success of PIM1 on the senescence of HCPCs, it augments the benefits of autologous transplantation with increased cardioprotective potential of already aged and diseased cells. When PIM1 is targeted towards nuclear or mitochondrial overexpression, it provides the option to individualize HCPC modification, providing a solution to the issue of patient-to-patient variability.
Scientists have already had success with regenerating cardiac tissue in mice in vivo and in human cardiac progenitor cells ex vivo. Now they're seeking to conduct human clinical trials where they obtain progenitor cells from the subject, modify them to overexpress the PIM1 gene, and then reimplant them into the heart. Ideally, this will cause the heart to repair its tissue. If the clinical trials lead to successful results, then this treatment can be implemented in hospitals in patients with degenerative heart conditions. One issue that may arise as a result is the formation of benign or malignant tumors. In conclusion, this approach to the treatment of cardiac degeneration can rejuvenate a heart and, in a sense, turn back the clock to combat senescence. PIM1 localization enhances the proliferation, survival, and youthful properties of aged HCPCs. It could potentially displace the heart transplant industry, which would mean avoiding the possibility of organ rejection by the body. Regeneration would be a much more natural option because the tissue would be generated from your own cells and therefore would not be considered foreign. The ability to target localization of PIM1 expression allows us to address patient-to-patient variability and design a more personal and specific treatment plan. Although we're not quite there yet, we are one step closer to the future of cardiovascular medicine.
We hope you enjoyed this video. Thanks for watching.